Okay, good morning, good afternoon, good evening from wherever you are listening. You are welcome to today's lecture video on chemistry of lipids. So by way of introduction, we want to highlight the previous lectures we had on the introductory portion of lipid chemistry. So what are lipids? Let's first look at the definition of biological lipids. We say that they are chemically diverse group of compounds which include fats, oil, steroids, waxes, and related compounds, and they are commonly defined by their feature of which is their insolubility in water, but they are soluble in non-polar solvents such as ether, chloroform, benzene, etc. So the biological functions of lipids is actually as diverse as their chemistry. We can say fat and oil are the principal store forms of energy in many organisms. We have phospholipids and sterols that are major structural elements of biological membranes. We are going to be discussing about the components of cell membrane, which lipids are the what major structural elements that are found in the lipid in the membrane. So the central architectural feature of biological membrane is a double layer of lipids which act as a barrier to passage of polar molecules and ions. So this architectural structure or feature of the what double layer of biological membrane allows it to regulate what goes into the cell and what comes out of the cell. We are going to be discussing that in our subsequent classes. So membrane lipids are also, they have what we call, they have hydrophobic portion and hydrophilic portion together, which they are termed as what amphipathic. It means one end of the molecule is hydrophobic and the other portion is what hydrophilic. We are going to be discussing all this in details in our subsequent classes. So first, let's look at what phospholipids are. We said phospholipids, they are lipids that contain, in addition to fatty acid and alcohol, they contain what we call phosphoric acid residue. They are they frequently have nitrogenous bases or nitrogen containing bases and other substituents. For example, you have glycerophospholipids, and you can also have sphingospholipids. In glycerophospholipids, the alcohol is glycerol, while in sphingospholipids, the alcohol is sphingosine. So you have to take note of the differences. In glycerophospholipids, the backbone alcohol is what? Is what? Glycerol 3-carbon alcohol. And sphingospholipids, the alcohol is a what? Sphingosine. We are going to see the structure of sphingosine. So many phospholipids may be regarded as derivatives of phosphatidic acid, in which the phosphate group is esterified with the OH of a suitable alcohol. So the OH group is esterified, right? The phosphate group is esterified with the OH of a suitable alcohol. We are going to see all the structure and all the components that will make us to understand the structures of the different structures of these lipids, different types of membrane lipids. So this structure below is what that of phosphatidic acid. You can see carbon that is labeled carbon 1, carbon 2, carbon 3. These are the glycerol carbons, 3 carbons of glycerol. And you have what? Carboxylic acid at position 1. substituted, and at position 2 you have another carboxylic acid, while at position 3 you have what? Phosphate group substituted. So this whole structure is called what? Phosphatidic acid. So phospholipids, this is their what? Backbone structure in which all other phospholipids are what? Derived. So phospholipids, they are what? Derivatives of phosphatidic acid. So this structure that you can see, you are seeing below, is phosphatidic acid, and all other types of phospholipids are derived from this. You can see the phosphate group that is attached to carbon, the phosphate group that is what? Attached to carbon 3. This is the phosphate group or phosphoric acid group, and you have negative, right? You have -1 here. You also have another -1 attached to the oxygen. So here is the point of attachment to other substituent groups to form different types of phospholipids. You understand, we are going to be discussing about them in our subsequent slides here in this lecture. So when you attach a substituent group here, you are left with a OH O negative here. Okay, so it means at neutral pH, the may tends to be what? Negatively charged because of this negative sign that is here on without forming any bond. So this is the point of attachment here. So let's look at this structure. You are already familiar with it. This is phosphatidic acid. Phosphatidic acid, in which you have 1 carbon, 2 carbon, 3. This is the glycerol backbone, right? And to carbon 1, you have what? Carboxylic acid. You have what? You have your what? Fatty acid here. This is fatty acid that is attached to it. And you know, fatty acid, they are what? Organic acid. So they have what? Carboxylic acid functional group. So this bond between the alcohol here and the what? Fatty acid, right, is called what? Ester linkage. So at carbon 1 and carbon 2, you have what? Ester linkage between the carbon 1 and carbon 2 of the what? Glycerol, and that's those of what? The fatty acid, depending on what is this R, how many carbons is this fatty acid? Is it short chain, long chain? Okay, is it saturated or unsaturated? You are all familiar with saturated, unsaturated, short and long chain fatty acid from your previous lectures. So, and also you have attached to carbon 3 the phosphate group, right? The phosphoric acid group. And this phosphoric acid group is now linked to the what? Glycerol in what? Phosphodiester linkage. So how many linkage do you have here? You have ester linkage and also phosphodiester linkage. Ester linkage in carbon 1 and 2, while in carbon 3 with the phosphoric acid group, you have what? Phosphodiester linkage. So another structure, this is the glycerol. You are all familiar with this glycerol from our previous lectures, from previous classes. You know that this is glycerol. So this is the backbone that was used to build the other substituent to form what? The phospholipids. So you have what? This as your glycerol. So this is carbon 1, carbon 2, carbon 3, right? This first carbon is esterified with a fatty acid, okay, in ester linkage. The second carbon 2 is esterified with another fatty acid with an ester linkage. And the third carbon, which is the third carbon of the glycerol, is what? Is attached to phosphoric acid in what? Phosphodiester linkage. So this structure is also important because we are going to be talking about different types of what? Phospholipids, and this choline is another substituent group that comes and attach itself to the what? Phosphoric acid group at this. Okay, so as we are discussing, sorry for the short breakout, we are discussing about this substituent group. We said that this choline is another substituent group that attach itself to the what? This blue portion where you have phosphoric acid O negative here. So this choline can attach itself to form another form of what? Phospholipid, which is what? Phosphatidylcholine. So this is ethanolamine that can also attach itself here. So if you have choline here, it's a different type of phospholipid, phosphatidylcholine. And if you have ethanolamine attached here, you have phosphatidylethanolamine, which is a different type of what? Phospholipid. So this phosphatidic acid is very important in that it plays a key role in cell structure. It also plays an important role in signaling and in metabolism. So it is a precursor to glycerophospholipids and also triglycerols. So let's look at what glycerophospholipids are. So glycerophospholipids, from the name, these are they are also called phosphoglycerides. They are membrane lipids in which two fatty acids are attached in ester linkage to the first and second carbons of glycerol, and a highly polar or charged group is attached through a phosphodiester linkage to the third carbon. So by now, when we talk about ester linkage, you are familiar with what ester linkage is. Let me share more light. When we say ester linkage or ester bond, this is a bond that is formed between from esterification reaction, and this esterification reaction is a reaction between an organic acid and an alcohol. So you have a linkage between the carbonyl group of the what? Let's say carboxylic acid, which is an organic acid. You have the carbonyl group of that carboxylic acid now forming a bond with what? The OH group or the oxygen group from the OH of a what? And of an alcohol. So that is what? Esterification reaction. So now the bond that is formed is called what? Ester bond or ester linkage. If you remember, you are let's go back small. If you remember this, this is our glycerol, this is an alcohol, and our fatty acids, they are what? Organic carboxylic acid. They carry out what? Carboxylic acid function. So this carbonyl group will now come and form bond with the OH group here. You understand? With the elimination of water molecule to form the what? This bond that is called the ester linkage. So for when that happens, you have what? Attachment of the fatty acid in ester linkage to the first and second carbon of the glycerol, and a highly polar charge group is attached to a phosphodiester linkage. So such as the what? In the case of what? The phosphatidic acid, right? You have first and second carbon fatty acid attached in ester linkage, and the third carbon you have what? Phosphoric acid phosphoric acid for attaching to the third carbon in what? Phosphodiester linkage. So glycerophospholipids, they are named as derivatives of the parent compound. What is the parent compound of glycerophospholipid? It is called phosphatidic acid. Now, phosphatidic acid, you are familiar with it. Now, according to the polar alcohol group in the head group, okay, according to the polar alcohol group that is found in the head group, that is the polar phosphoric acid functional group that is found. So for example, if you have phosphatidylcholine and phosphatidylethanolamine. Now for phosphatidylcholine, you have choline attached to the what? Phosphoric acid. While in phosphatidylethanolamine, you have what? Ethanolamine attached to the what? Phosphoric acid group, which is the what? The polar head group. You understand that. So in all these compounds, the head group is joined to glycerol through phosphodiester bond. Okay, the head group is joined to the glycerol in phosphodiester bond. We have explained that. Now, in which the phosphate group bears a negative charge. Now, a phosphate group bears a negative charge at neutral pH. I have explained that. Now, the fatty acids in glycerophospholipids can be any of the wide variety of fatty acid that you are familiar with. And in general, glycerophospholipids contain 16 carbon to 18 carbon saturated fatty acid at carbon number 1. So at carbon 1 of the glycerol, the glycerophospholipids contain 16 or 18 carbon saturated fatty acid. You know, 16 carbon saturated fatty acid is what? Palmitic acid. 18 carbon saturated fatty acid is what? Stearic acid at carbon 2, while at carbon 1. Now at carbon 2, you can have 18 carbon or 20 carbon unsaturated fatty acid at carbon 2. 18 carbon or 20 carbon unsaturated fatty acid. You have 3 different types of 18 carbon unsaturated fatty acid. So there is one with 1 unsaturation, there is another one with 2 unsaturation, and there is another one with 3 unsaturation. So by now you must have heard about oleic acid, linoleic acid, and linolenic acid. Oleic acid, 18 carbon unsaturated with 1 unsaturation, 1 double bond. Linoleic acid, 18 carbon, 2 double bonds, while linolenic acid, 18 carbon, 3 double bond. So the common glycerophospholipids that are here listed are some examples of the common glycerophospholipids, which include phosphatidylcholine. In other words, it can be called lecithin. So when we say lecithin, we mean what? Phosphatidylcholine. Then there is phosphatidylethanolamine, which is called cephalin. When we say cephalin, we means what? Phosphatidylethanolamine. We have phosphatidylserine, we have phosphatidylglycerol, and phosphatidylinositol, and we also have what we call cardiolipin, and this cardiolipin is also known as what? Diphosphatidylglycerol. And lastly, we have plasmalogen. So we are all going to be discussing all these phospholipids. All these phospholipids, we are going to be discussing them one after the other, and we are going to be explaining them. So now let's look at phosphatidylcholine. Now for phosphatidylcholine, they are a class of phospholipids that have choline. They have choline as their polar head group. And you have already seen the structure of choline. I have introduced you to the structure of choline. So they have this choline as their polar head group. So it means if you look at this structure, it means at this position you have choline attached to the polar head group. Okay, and they are the most abundant phospholipid of the cell membrane, and they represent a large portion of the body store of choline. And this choline is very important in normal health of the organism and also in the nervous system, and it also serves as the storage or reservoir for what we call the labile methyl groups. And these labile methyl groups, as you can see, it has methyl groups here. It has 3 methyl groups, so it serves as like storage of labile methyl groups that are very important in normal cell and body functioning. So here we have R1 and R2 to be the acyl carbon chain, that is the carbon chain from the the fatty acid group, fatty acyl group that are attached at carbon 1 and carbon 2. So this is phosphatidylcholine. Now, choline, as we explained earlier, it's an important component in nervous transmission. It helps in communication, nervous transmission, and also it is in the form of acetylcholine, and it also serves as store form of labile methyl groups. That is, this lecithin is also called dipalmitoyl phosphatidylcholine. It means 2 palmitic acid, right, at carbon 1 and carbon 2. You remember, we say depending on the number of carbon, we can have a carbon 16 or carbon 18 saturated at carbon 1 and carbon 2, right? So this is dipalmitoyl 16 carbon palmitic acid at carbon 1 and carbon 2. So, and you have choline at carbon 3 attached to the phosphoric acid or head group. So you have dipalmitoyl phosphatidylcholine, which is lecithin, is very important component of what component of the lungs, okay, and it is important in that it's effective surface active agent, and it is a major surfactant that is preventing adherence due to surface tension of the inner surface of the lungs. So it is found in the lungs, and it helps as what surfactant that prevent what surface reducing surface tension in the lungs, so as to allow the organism to breathe normally. So in the absence of this lecithin from the lungs of premature infants, it causes what we call respiratory distress syndrome, RDS. So this is one of the components, one of the substance that have been assayed for. They have been checked during infancy or during pregnancy before delivery. The lecithin can be measured. Lecithin, I think lecithin myelin ratio can be assessed from checking the sample of amniotic fluid to know whether an infant is healthy or is at risk of developing this respiratory distress syndrome. So for phosphatidylethanolamine, we call it cephalin. It is usually the second most abundant phospholipid that is found in animals and plant lipids. Now, after phosphatidylcholine, because phosphatidylcholine is the most abundant, and phosphatidylethanolamine is the second, and it is frequently the main lipid component of microbial membranes. Phosphatidylcholine differ from phosphatidylethanolamine in that in phosphatidylcholine, ethanolamine is replaced. In phosphatidylethanolamine, ethanolamine replaces choline. So if you remember the structure of phosphatidylcholine, you remember this is where we have the choline attached. So in the case of phosphatidylethanolamine, ethanolamine is attached here at the phosphoric acid head group. So this is a phosphatidylethanolamine structure. It acts as a chaperone which help the membrane protein to fall into their tertiary structure so as to function properly. That is one of the most important function of phosphatidylethanolamine. So we have what we call phosphatidylserine. Now, phosphatidylserine is a glycerophospholipids that is found in tissues which consists of two fatty acids that are attached in ester linkage to the glycerol, and also you have serine attached through a phosphodiester linkage to the third carbon of the glycerol. So if you look at this, the two carbon 1, carbon 2, you have fatty acid attached in ester linkage, and the third carbon, you have this is serine now attached in phosphodiester linkage. Now, this is the structure of phosphatidylserine, and it plays an important role in cognitive function and in cell signaling, especially in apoptosis. And it also required for healthy nerve and cell membranes and myelin function. So when we say apoptosis, apoptosis is a programmed cell death, okay, and the body tries to get rid of cells in a kind of regulation, okay, in order to maintain normal function. So how does we have what we call phagocytosis or phagocytes, which are some special white blood cells, the macrophages, the neutrophils, right? So they are phagocytes that helps in engulfing foreign, let's say, microorganisms when they embed or a cell that is going to undergo apoptosis. So they engulf it, okay, in a process that is called phagocytosis. So how do they know which cell to engulf and which one not engulf. So is this phosphatidylserine that now show itself. Remember, they are membrane lipids. So it now show itself and like a signal in the cell membrane. It will now show itself at the cell membrane. When the phagocyte signal sees that signal of phosphatidylserine protruding from the cell membrane, it means this cell should undergo apoptosis, and now phagocyte will now carry it and what phagocytosis and takes place. So, and also they are also important in nerve cell membrane and myelin. When we say myelin is, we remember we are going to be discussing phosphatidyl. We are going to be discussing sphingomyelins, and these are like they serve like an insulator to the what nerves. This myelin, we have what we call myelin sheet, and this myelin sheet serve like an insulator to the nerves, right? So they are also phosphatidylserine are also important in myelin function. So we are going to stop here, and we hope to meet and continue with the subsequent types of what phospholipids and also other components of these membrane phospholipids. So thank you very much for listening. And if you are yet to subscribe to Z-Town TV 995, you can do that. And I hope you enjoyed watching the videos that we are bringing to you. So subscribe, you can share, you can like, you can comment. If you have any question, you can drop it in the chat box. And if you have any concern, you can also drop it there. So thank you very much. We are going to stop here and see you in our next class. Bye-bye.